STB1093 Microbiology. Experiment 4. Staining Microorganisms.
Enhancing the contrast between cell and background is achieved through the utilization of stains that undergo chemical reactions with cellular material. The method of simple staining involves the application of single stain. The stains incorporate chromophore that chemically react with specific cellular components or structure, enabling the visualization of various unique microstructures such as flagella, endospores, and cytoplasmic inclusions. Differential staining has been developed for more detailed information. The stains exhibit varying reactions with different cell types and play a crucial role in identifying taxonomic groups. One of the most significant and widely employed forms of a differential stain for bacteria is the Gram stain. Based on the response to the Gram stain, bacteria can be categorized into two major groups, which is Gram-positive microorganisms and Gram-negative microorganisms. The Gram stain incorporates the principles of a simple staining and serves to distinguish bacteria into two main categories, Gram-negative microorganisms and Gram-positive microorganisms. Gram stain techniques get its name from the Danish bacteriologist Hans Christian Gram, who first introduced it in 1882. His initial aim was to differentiate all bacteria from mammalian tissue. It took another year for Gram to discover that other bacteria also decolorize quickly, leading to the realization that he did not achieve his initial goal. In the Gram stain procedure, there are four reagents used, which are crystal violet, iodine solution, alcohol or acetone, and safranin. The decolorization step holds significant importance in the procedure. Gram-positive microorganisms resistant to the decolorizing agent retain the primary stain, rendering them purple in color post-staining. Conversely, Gram-negative microorganisms lost the primary stain during decolorization and upon counter-staining with safranin exhibit a pink hue. In the absence of iodine-mordant treatment after primary staining, Gram-positive cell may also lose their primary stain during decolorization, mimicking Gram-negative cell. Several factors that can affect the result of the Gram stain procedure. The smear preparation, concentration and freshness of reagent, consistency of washing and drying the smear between steps, and reliable of Gram stains only on cells from cultures that are in the exponential phase of growth.
Are prepared from cultures of two distinct microorganisms, bacteria A and bacteria B, representing each of the two groups. These smears are air dry and heat fixed. Next, the slides are positioned on a staining rug and each smear is inundated with crystal violet. After allowing the stain to set for 1 minute, the excess crystal violet is rinsed off from the slide using tap water and excess water is drained. Following this step, each smear undergoes flooding with iodine for 1 minute, which is subsequently washed off using tap water. Tightening each slide, decolorization is carried out using 95% ethanol until the draining alcohol appears colorless, typically taking around 50 seconds. The slides undergo a brief wash with tap water to remove any remaining ethanol and excess water is drained. The smears are counter-stained with safranin for about 20 to 30 seconds, followed by a brief wash with tap water and blood drying. Finally, the slides are examined under the oil immersion objective, aiming to identify cell displaying either Gram-negative or Gram-positive appearance.
This is the result obtained after viewing the microorganisms under the microscope. Escherichia coli is a Gram-negative bacteria. When viewed under the microscope, it was rod-shaped. It has a thin layer of peptidoglycan cell wall and it was about 1.5 micrometer long. There is also a presence of flagella. Next, Staphylococcus aureus is a Gram-positive bacteria. It is round in shape and it occurs as clusters. It has a very thick layer of peptidoglycan cell wall and about 0.5 to 1 micrometer in diameter. It is also lack of cilia and flagella. Discussion. When slide A is viewed under microscope, a slight pink to purple colored rod-shaped bacteria are observed. This is because they lose their crystal violet iodine complex during the decolorization step. However, since they absorbed the counter stain, which is safranin, they turned pink. purple in color. This is a gram-negative bacteria. This is supported according to Cleveland Clinic 2022. When exposed to a gram stain method, gram-negative bacteria exhibit red to pink coloration due to their thin peptidoglycan layer of cell walls and high lipid content. However, as we can look at the picture beside, the stain that we obtain from this slide is not really red to pink in color, but more to pink to purple color. This is because the bacteria were exposed to the counterstaining reagent, which is safranin, for an extended period of time, which results in a deeper, more purple colored cells. Let's look at the discussion of slide B. When slide B is viewed under microscope, a dark purple colored round-shaped bacteria were observed. These are gram-positive bacteria as they retain the primary stain and remain in purple color. Therefore, this is a Staphylococcus aureus bacteria. It has a very thick peptidoglycan cell wall which takes in purple dye when gram-stained. So, this is a gram-stained bacteria. Besides, electron micrographs shows that the peptidoglycan to be 20 to 80 nanometer thick. In gram-positive cells, the peptidoglycan accounts for 50% of the cell weight and 90% of the weight of the cell wall. However, as we can look at the picture beside, there are only few colonies of S. aureus present. This may be due to the inoculating process was not uniform, or there was an uneven distribution of bacteria on the slide when smearing it. Let's look at the questions now. Question number 1. If the iodine step were omitted, what color would you expect a gram-negative microorganism to be? A gram-positive? The gram staining procedure would not be complete without the iodine step, which acts as a mordant and helps to fix the crystal violet dye in the bacterial cells. The crystal violet dye would not be effectively trapped in the thin peptidoglycan cell wall in the gram-negative bacteria without the iodine step. As a result, during the subsequent step, the alcohol would wash away the crystal violet from the gram-negative bacteria, which results in pure retention of the primary stain. Therefore, the gram-negative bacteria may appear lighter or less distinct in color. However, they are still considered a gram-negative bacteria and not gram-positive. Let's look at the second question. Question number 2. Could other dyes be substituted for crystal violet in the gram stain procedure? And for safranin, explain your answers. Yes, indeed, other dyes can be substituted for both the crystal violet and also safranin in the gram stain method. For example, methylene blue is a basic dye that has a blue color, which can substitute the crystal violet in the gram stain. It works just the same as the crystal violet in the gram staining procedure. As for safranin, neutral red can be substituted. The gram-negative cells are similarly stained by neutral red, which gives the required contrast. Question number 3. What role does the mordant play in the gram stain procedure? First thing first, in a gram stain method, the mordant is typically a solution of iodine. The main function of it is to enhance the staining process by forming a complex with the crystal violet dye. This is an important step as it helps to distinguish between gram-negative and gram-positive bacteria. Let's look at the last question. What correlation is there between cell wall composition and gram reaction? Is gram reaction correlated with sensitivity to various antibacterial agents? Regarding sensitivity to antibacterial agents, there is a correlation between gram reaction and susceptibility to certain antibiotics. For an example, gram-positive bacteria tend to be more susceptible to antibiotics that target the peptidoglycan layer, such as the penicillin. This is because it has a very thick peptidoglycan layer that provides a target for these antibiotics to disrupt cell wall synthesis. At the same time, gram-negative bacteria are also resistant to certain antibiotics due to the presence of the outer membrane that acts as a barrier. This outer membrane can prevent the entry of certain antibiotics, making the gram-negative bacteria inherently less susceptible to some classes of antibiotics.